All right, we're on the home stretch, chapter 17. I'm actually not going to cover everything in this chapter. It's already kind of a short chapter, but we're not going to cover everything in it. You're welcome. We're going to cover the important bits. But what I want you to get out of this chapter is I want you to get at least a basic understanding of how genes actually encode for observable phenotypes. So, one thing that we looked at when we were first looking at Mendelian genetics, right? We were looking at a gene that encodes purple and white flowers. So the reality is that the gene that you were looking at encodes a protein, and that protein is an enzyme that can synthesize the purple pigment that turns the flowers purple. And that's just one example of how gene expression works and how genes can encode observable phenotypes. But at the base of this concept is this idea of genes encoded in DNA ultimately encoding proteins, which do all the different things inside of a cell. So genes encode polypeptides, and the polypeptides are what do the things associated with that gene, right? So for the purple flowers, the polypeptide is what made the purple pigment that turned the flower purple. There's, I'm sure, in the fruit flies that you were looking at with the white eyes and the red eyes, the ones with the red eyes have a similar phenomenon happening with some kind of enzyme, probably making a red pigment that turns the eyes red, and that's just absent in the white-eyed flies. So, in a very broad sense, the process looks something like this. You start with DNA, right? And the DNA has genes encoded in them. And then that DNA is used as a template to make what we call mRNA, messenger RNA. This process right here is called transcription, right? A transcript of that gene in the DNA is made. The transcript is RNA. So basically what will happen is a copy of the DNA gets made, but it's just an RNA copy of the DNA gene. And then that RNA copy is used as a blueprint to make the polypeptide. That process is called, we already learned about it, translation. We've already talked about this, not much detail, but we did touch on it. Translation is just the process by which an mRNA transcript is used as a blueprint to make a polypeptide or a protein. And so we're going to start here. We're going to start with transcription and look at it in a very general sense. All right, so if you look at a gene, right, it's encoded in DNA. DNA is this double-stranded, try that again. We'll draw double-stranded DNA, 3', 5', and then over here, 5', 3'. So for every gene encoded in your DNA, there's a double strand of DNA, right? Only one of these strands actually encodes the gene. So one of them gets used as a template and the other one doesn't get used as a template. So for this particular one, we'll be looking at this top strand is what we'll call the template strand, and the bottom one is the non-template. All right, and in this example, the direction of transcription is going to go this way. So if transcription went the other way, then this one right here would be the template, and this would be the non-template. And this is because RNA polymerase, just like DNA polymerase III, RNA polymerase can only add nucleotides to the 3' end. Pretty much any time you're extending a nucleic acid strand, the new nucleotides are going to be added to the 3' end. So if this top strand were being used as a template, right, then our RNA, which we'll do in red, all right, the RNA strand would be 5', 3'. So the RNA strand gets extended in that direction. So transcription, just like DNA polymerization, goes 5' to 3'. Anytime you're looking at nucleic acid extension, you're looking at 5' to 3' extension. So in this case, this top strand is being used as a template to make the new RNA strand. So that's why it's the template strand. We're going to use bacterial transcription as our example here because it's a little bit simpler. We're not going to look at the details of eukaryotic transcription. So let's say here's our template strand. All right. And transcription is going to go in this direction. This is our DNA strand. There's a region of the gene called the promoter region. And the promoter region is just a region of the gene where RNA polymerase can bind to the gene. So this is RNA let's try that again. RNA polymerase. So RNA polymerase will bind to the gene at the promoter, and then it will just extend along the gene. It'll just travel along the gene, creating a new mRNA strand. And it'll just kind of spit that RNA strand out the top. And eventually it will get to the end of the gene and it'll break off and it will release the RNA strand that it just made. Right? So just to recap, RNA polymerase will bind to the double-stranded or the single-stranded DNA at the promoter region, and then it will travel along that single strand of DNA, creating an RNA strand that is complementary to that DNA strand. And then when it gets to the end of the gene, it'll break off, and it can go on to bind to another gene and make another transcript, and it will release its RNA strand. And so that RNA strand can then get used as a blueprint for translation. All right, and we're going to skip eukaryotic transcription. But I do want to point out some differences between eukaryotic and bacterial RNA processing. So in bacteria, it's, as with a lot of things, it's just a lot more simple, right? So DNA gets transcribed to make RNA. And then that RNA can just go straight to translation. Right? So the DNA gets transcribed, right? And so a copy of the DNA is made, an RNA copy of that DNA is made, and then that RNA copy can go straight to a ribosome, and it can be used as a blueprint to make protein. By the way, I want to go ahead and nip this in the bud because there's a mistake that people make every semester, no matter how many times I could stand up, jump up on a table, wave my arms, and say, don't make this mistake. People will still make the mistake. Don't look at this and think that DNA gets turned into RNA. That doesn't happen, right? Don't say that DNA gets transcribed into RNA. It doesn't become RNA, right? An RNA transcript is made using the DNA as a template, right? And kind of along the same vein, don't say that RNA gets turned into a protein by a ribosome. Again, it's not what happens. DNA or RNA doesn't turn into a protein. The RNA is used as a blueprint to make the protein. So, somebody's going to make that mistake. Don't let it be you. So, yeah, I just want you to I want to make sure that you have that straight in your head. The DNA stays DNA, always and forever. It will always be DNA, right? It's just used as a blueprint to make a copy, an RNA copy, a transcript. Okay, anyway, with bacteria, it's pretty straightforward. That RNA, once it's made, it can just go straight on to translation without any kind of modification. But in eukaryotes, it is a little bit more complicated. In eukaryotes, all right, it starts the same. DNA, it's transcribed. To make an RNA copy. All right. We call this we'll call it pre-mRNA, right? Because it needs to be modified before we can consider it mRNA. So it has to go through processing, which we'll detail before it can become mRNA. And this processing happens in the nucleus, right? So the DNA gets transcribed in the nucleus to make pre-mRNA. That pre-mRNA gets processed in the nucleus still to make mRNA, and then that mRNA exits the nucleus, and now it can go on to translation. Now it can be used in translation. So there's a few steps that have to happen to the RNA before it can be used in translation. mRNA, by the way, the M stands for messenger. Think of it like it's a messenger that's going to travel from the nucleus out into the cytoplasm, and it's taking a message from the nucleus where the activities of the cell are being controlled. This messenger RNA carries that message from the nucleus out into the cytoplasm where it affects what proteins are being expressed. So that's why it's called messenger RNA. So let's talk about that mRNA processing. Eukaryotic mRNA processing. All right, I'm just going to list them for you, and then we'll detail what it actually means. So there's three things that happen. One is called splicing. Another is called a 3' polyadenosine tail. And then you've got a 5' cap. All right, so these are the three processing steps, and we'll go into a little bit of detail about each one of them. And we'll start with splicing. If you look at a strand of pre-mRNA, it looks kind of like this, right? Let's say here's your strand, 5', 3'. There is a region, let's see, I'll label it in red. There's a region right here on the 5' end called the 5' untranslated region, or UTR. All right, this is, just like the name would imply, is the region at the 5' end of the mRNA that doesn't get translated. It's the untranslated region. And then you've got a long stretch called the coding segment. This is the segment that actually encodes the protein encoded by this gene. This 5' UTR doesn't encode any protein. The protein is all encoded in here. And then there's also a 3' untranslated region. Right here in the coding segment, there are sequences that are translated and sequences that are not translated. Right? So there's two types of sequences here. They're called exons and introns. So we'll denote them like this, right? Where it gets thick, that's an exon, and where it's just a line, that's an intron. So some of these exons are long, some of them are pretty short, some of them are very short, and then some of them can be really long. So this encodes part of the protein, this exon encodes part of the protein, this exon encodes part of the protein, and then this exon encodes part of the protein. These introns right here, there's an intron right there, there's an intron right there, there you go. The introns are just gobbledygook. They're just nonsense. They don't encode any protein at all. And so these introns need to be removed, right? Before this pre-mRNA is ready for translation, the introns have to be removed. So the splicing removes the introns. Right? It splices out the introns and connects all the exons together so it encodes one continuous protein. If you look at the first slide, what you're looking at is a gene. All right? And the gene encodes a bit of pre-mRNA that can be modified to become mRNA. And in that pre-mRNA, there are introns and there are exons. And the reason I'm showing you this is just to give you an idea of how big these different elements are. So in this particular sequence, there are red letters and there are black letters. The red letters are encoding introns, and I'm sorry, did I say the black? The black letters are encoding introns, and the red letters are encoding exons. So what I want you to see is that there's a lot of variability in the lengths of these various sequences. Some of the exons are really short, some of the exons are really long, some of the introns are huge. And so before this piece of pre-mRNA is ready for translation, all those introns need to be removed so that you have one continuous sequence that encodes your protein. And that's actually, by the way, that's actually a real gene. It's called Sonic Hedgehog. And this is just, one of the cool things about science is that when you discover something, you can name it whatever you want. And if you're real pretentious and you think real highly of yourself, you can name it after yourself, right? Barbody's a guy named that after himself. Krebs cycle, a guy named it after himself, right? You can name it after anything you want, though. You can name it after your dog. You could name it after your favorite video game character, right? Sonic the Hedgehog is actually named after Sonic the Hedgehog. So you can name it whatever you want. You have a lot of freedom. Right? So the splicing just removes all the introns and sticks the exons together so that they encode one continuous protein. The next slide. Yeah, we're not going to worry about that. We're not going to worry about alternative splicing. You can worry about that when you take genetics. All right, so that's the splicing. All right, that's why we need splicing. You've got to remove these introns so that the exons are stuck together. The other two mRNA modifications happen at either ends of the mRNA strand. So at the 5' end, you've got a 5' cap, and at 3' end, there's a poly A tail. So the 5' cap is just a modified guanine nucleotide. And what that does is it protects the RNA strand from degradation. There are enzymes called RNase. As the name would imply, these are enzymes, ACE, that chew up RNA. And so the 5' cap protects that messenger RNA from these RNA degrading enzymes that can chew it up. All right? At the other end of the mRNA, you've got a 3' polyadenosine tail. And as the name would imply, it's just a really long chain of adenosine nucleotides. On the 3' end. And you can kind of think of it as, we'll call it an expiration date. Think of it this way. All right, let's say a piece of mRNA gets made in the nucleus. And let's say it gets sent out into the cell to get translated to make protein. It wouldn't be very helpful if that mRNA strand just stuck around forever, right? If the nucleus sent out that messenger RNA, you know, ostensibly, the cell doesn't need to make that protein forever, right, indefinitely. The cell needs to be able to control the expression of all the proteins that are being made. So all the mRNA that gets sent out of the nucleus has an expiration date. And it really just depends on how long that tail is, because you got all these RNase enzymes that will chew up the RNA strand at the 3' end. And so those RNase enzymes will just chew up that poly A tail, and then eventually they're going to start chewing up the part that actually encodes the protein, and at that point the protein stops being made. So that's the function, that's the benefit that this poly A tail serves. So once the mRNA gets sent out of the nucleus, that poly A tail will start to shorten and shorten and shorten and shorten and shorten, and eventually the mRNA just gets chewed up and it stops getting translated. So this works because the mRNA is degraded from the 3' end in the cytoplasm. And eventually the poly A tail gets chewed up, and then the coding part of the mRNA gets destroyed as well. All right, so once the mRNA leaves the nucleus, and it can't do that until it's had all three of those modifications done. It has to have splicing, it has to have the 3' tail and the 5' cap. Can't leave the nucleus until all that happens. But once it goes out into the cytoplasm, then it gets translated. And translation, once again, is just the process by which that mRNA strand is used as a template to make a polypeptide. And So there's only 4 nucleotides, but there are about 20 or so amino acids. So it's not just a simple one-to-one translation. It's not like one nucleotide can encode one amino acid. Because there's so many more amino acids than there are nucleotides, those amino acids have to be encoded in groups of 3 nucleotides, and we call those groups of 3 codons. So a codon is just a nucleotide triplet that encodes a single amino acid. I'll do it singular. A codon is a nucleotide triplet that encodes one amino acid. And if you look at the next slide, right, the one that says genetic code, and then there's a table underneath it. That's the table of codons and all of the amino acids that they encode. So what you see there is, in the top left corner, right, you see 4 codons in that top left box. There's UUU, UUC, UUA, and UUG, right? These are 4 different codons, and you can read this table just by finding, if you want to know what any codon encodes, you just look at the codon. Let's say it goes AUC, right? And you want to know what amino acid is encoded here. Well, the first one is A, so you look on the left side of that table. It says first letter, and you find A right there on the left side. And then the second letter is U, so you go over to the second box, right, because that's where it goes UA. And then within that box you find UAC, right? And UAC encodes isoleucine, I-L-E. That just stands for isoleucine. You don't need to know what all the abbreviations are for the amino acids, but you do need to know how to read a codon table, right? So what you need to be able to do is look at a sequence of nucleotides, and you need to be able to tell what the sequence of amino acids is that is encoded by that nucleotide sequence. So for example, let's say you've got a sequence of nucleotides that goes, let's see, G, U, A, U, G, C, 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 G, A, C, G, C, hold on a second. Make sure I get this right. I'm freehanding here. Okay. And of course, these are red 5' to 3'. Put a big fat star next to this. Write this down. mRNA is translated 5' to 3'. All right, so in order to read this mRNA strand, right, this is mRNA, you have to know where to start reading, because there's 3 frames, right? You could read within, right, you could start here, in which case it would go GUA, that's one codon, UGC, CCG, ACU, etc., right? Or you could start here and go UAU, GCC, CGA, right? That would give you a totally different protein. And then you could also start here, and it would go AUG, CCC, GAC, and that would give you a totally different protein. So because codons have 3 nucleotides in them, there are 3 reading frames that you could use to read this mRNA strand. So how do you know which one to use? Well, conveniently, all polypeptides begin with methionine. They always start with methionine, which on that table right there is abbreviated MET. That stands for the amino acid methionine. And so all the polypeptides are always going to start with methionine. That's why methionine on that table is in red. That's what we call a start codon. So every polypeptide is going to start with a methionine amino acid. And the codon that encodes methionine is AUG. All right? So, if this strand of mRNA was being translated, it would start with an AUG. So the ribosome might bind on to this mRNA strand and start reading. And when it sees an AUG, it will start translating. So there's our start codon. So the first amino acid in this polypeptide is AUG. Net, methionine. The next one is CCC. The next codon is GAC, then UGC, then UGA, go dogs, and UCC. I'm actually a UAB fan. I don't care about UGA. But that is an important codon. We'll get there. All right. So the first amino acid that gets made is methionine. Next one, CCC. All right. I would hit pause on this video and see if you can translate the rest of this mRNA strand. So go ahead and try it on your own. Pause. All right. So you're back. Let's see if you got it right. CCC encodes proline, P-R-O. GAC encodes asparagine, or no, aspartate. Yeah, ASP. You just need to know, you just need to get these 3 letters right. You don't need to know what they mean. UGC, what is that? UGC, oh wait, where is it? Cysteine. CYS. UGA. Well, I already know what that one is. You might have figured this out, right? It's a stop codon. Stop is not an amino acid. Stop is what you do when you get to a stop codon. Stop is what the ribosome does when it reaches a stop codon. So the polypeptide encoded by this very short chain of mRNA is only 4 amino acids long. It goes methionine, proline, aspartate, cysteine. That's the end of it. Stop is not an amino acid. Write that down. I'd write that down on that table. Big fat capital letters. Stop is not an amino acid. It's what the ribosome does when it reaches a stop codon. Yeah, and so that's it. So that's the end of the show. I'm not going to talk about Yeah, I'm not going to talk about transfer RNA, not going to talk about ribosomes, not going to talk about all that. Yeah, y'all get off easy. So that's all you need to know about anything for the rest of your entire life, right? You don't need to know anything else, at least for this class. All right, I will see you, I guess, in the fall, hopefully.